Okay. I'm pleased to introduce Iris Kennedy, who is our 2022 Maxwell Hammerhand intern in the plant ecology program. Iris grew up in Shippensburg, Pennsylvania, and graduated from the University of Virginia in 2022 with a BA in biology and a minor in environmental studies. Her experiences prior to coming to Archbold include work on invasive buckthorn removal and an internship with the Environmental Defense Fund. Iris arrived at Archbold in late May of last year. As you'll see, her talk is titled Sifting for Treasure, but I'd say that the real treasure is Iris. Thanks. Thanks so much. She's been wonderful to work with and a key member of our team. As you'll see, she's come to be very fond of seeds and was the lead on our annual seed collections, in addition to all the work that you're going to see today. She was also a member of the DEI committee and wrote a wonderful newspaper article while she was here. After hours, she's an expert salsa dancer and instructor who is known around here for holding lessons for the other interns and staff. She's also a pretty decent artist who has done some wonderful drawings of many of our scrub species. Iris will be starting a new position this spring with the Smithsonian Institute in the forests of Northern Shenandoah, conducting tree surveys for the Forest Geo Project. And with that, I'll hand it over to Iris.
Hey, thank you so much, Erin. Hi, everybody. I'm Iris, and today we are going to go sifting for treasure. So let's go. To start off, I want to talk about soil seed banks. So this is a really important part of the life cycle for a lot of plants. Basically, so plants form their seeds and they drop those seeds. And instead of immediately germinating into seedlings, the seeds will go into the sand and just stay there dormant. And then eventually they'll receive some sort of trigger to germinate. So this is a really important strategy for our scrub plants because it allows them to be resilient to things like fire that will destroy above ground populations or above ground vegetation. And then the seeds will germinate out of the seed bank and replenish that population. It's also a very important reservoir of genetic diversity for plants. This figure down below, we don't need to go into all the details of it, but I just wanted to give a little sneak peek of the life cycle of the plant I'll be talking about and showing that it has all these important stages, but they all sort of feed into the seed bank. It's a very important aspect of that plant's life cycle. So here is the magic plant of the day. It is called Eryngium cuneiform. Normally I would talk about it using its common name, but for Eryngium, it's called wedge leaf button snakeroot, which is way too long in my opinion. So I'm just going to call it Eryngium today, but it's one of our rare endemic plant species. It was declared as endangered in 1987, and it's actually a member of the carrot family. So if you ever find some in the scrub and you want to smell it, it smells really good. But Eryngium relies on fire to create these clear gaps for it to grow. It sprouts best in bare sand. And I don't know why it's shaking like that. Okay. Train coming. So yeah, it sprouts on this bare sand, and we usually find it in rosemary bulb habitats, which have those big white sand gaps. And the most important thing about Eryngium for my project is that it forms a persistent seed bank. So it drops seeds into the soil and they stay there. As you can see from this picture, it has a very distinctive wedge leaf pattern. These are juvenile plants, and we have the flower structure on the right, which, fun fact, is a host to the black swallowtail butterfly, which I got to see a lot of while I was out doing my sampling. So on to another fascinating thing, roadside. So when I first got to Archbold, I noticed, and probably most of you have noticed this as well, our sandy fire lanes, which I'm just going to be calling roadsides for the rest of my project, but they often are host sites for tons of plant diversity. And often in the case of Eryngium, there's a bunch of rare endangered species just growing on the road. And that really stood out to me when I got here. And so got me thinking are there actually seed banks in these roads as well? Or is it just there's seeds falling in the road and they're instantly sprouting into plants? So that question really takes me into my project. This is just proof that Eryngium really thrives on the roads. These are a couple of little seedlings that I found popping up on the road. So the goal of my project was broadly just to characterize these Eryngium seed banks. The overarching question is like, how are these natural populations comparing to the roadside populations? So the Eryngium growing in the rosemary bulb, what do those seed banks look like when you compare it to the potential seed banks in the road? I also wanted to look at the effects of vegetation. So for this one, I looked at, does Eryngium cover predict what the seed bank will look like? And also, does the proximity to rosemary affect the seed bank? And why I wanted to look at rosemary was because rosemary is allelopathic, which means it has the ability to suppress plants from growing. So I thought that could maybe have an impact on where seeds were building up if they're being suppressed. Lastly, I just wanted to do a detailed dive into the roadside and how does that affect the spatial patterns of seed banks. So on to the methods. Thank you, Julie, for making my field maps for me. I chose 4 interior sites. So I went to 4 different rosemary bulbs, and then I also selected 1 roadside site where I went really like in depth and did like 37 transects. And I'll talk about this more later, but the reason I chose this roadside site is because the plant lab has been doing demography at that site for quite a while. So being able to map my data to demography data is going to be a key component of this project. So for the rosemary bulb sites, as I mentioned, I went to 4. I have 3 populations where Eryngium is thriving. And then I also had 1 like historic population where it's now extinct above ground. And I was just curious, like, to see if the seeds were still in the soil there. And I set up 10 plots in each rosemary bulb. So I randomly selected rosemary, and then I had these 2x1 meter PVC transects that I laid out, and I chose to put the near distance 0.5 meter away from rosemary, and the far distance was 2.5 meters from rosemary, because I read a paper that mentioned like a scrub or a shrub within 2 meters can sometimes have an effect on the germination of Eryngium. So at each of those near and far distances, I took 3 soil cores to a depth of 8 centimeters. And these are pretty big soil cores. I wish I remembered the like circumference or the volume of it, but I'll show that in a bit as well. And in each plot, I tried to do half of my, or in each rosemary bulb, I had half of my plots with Eryngium present and half of them without, so I could compare the effects of Eryngium cover. For the roadside, this is a breakdown of bulb 1, so the plant lab's demography site. What we have here is the scrub line is at the bottom and the road is at the top. And thankfully, plant lab has already like mapped out where to put the transects, so they made my life a little easier. I just added my flags in there. But basically from 0 meters to 180 meters down the road, I set up transects every 5 meters. And then at each of those transects, I measured out the distance from the scrub to the road. So I went 1, 2, 3, and 4 meters. And 4 meters took me not quite into the tire tracks, but pretty close to the road. And then I took soil cores at each of those distances. So with this setup, I wanted to get at, both the direction this way of like how Eryngium was spreading, because we have a theory that like the population here started a while ago, sort of near the 0 meters, and it's sort of invading down the roadside. But I also wanted to characterize it in this Y direction. So looking at, the scrub to road transition, as well as maybe the curvature of the road and how that, could be affecting seeds rolling or pooling up in certain areas. For my sampling and processing, so this fancy PVC tool is what I use to do my soil cores. Lupe Gonzalez cut it for me. I just needed something that was big, but also very light because I was carrying a lot of sand, and this was a perfect tool actually. So I went to a depth of 8 centimeters. This is an image of my roadside setup. It's a little hard to see, but I would like put my bags at each of the 4 distances and then take 3 cores, put them in a bag, 3 cores, put them in a bag. And I did this all the way down the roadside. It was a similar setup for the rosemary bulb. I would just only have 2 bags for those. Put it in the bag. And this is how I did a little bit of like preliminary depth analysis just to make sure I was capturing all of the seeds. I looked at the depth from 0 to 4 centimeters and 4 to 8 centimeters. So then I took all my bags of sand and put it all over the counters in plant lab and took up all of our space and just let them chill there and dry out. And eventually I poured them through a series of sieves and then the contents of the sieves I poured out onto a white sheet of paper. And then we find our treasure. So the reason Eryngium, or one of the reasons Eryngium is so great for this project is that you can just see the seeds by eye. Like you don't have to do microscopy or like germination trials. It was just me and some tweezers picking through all of my samples, which is tedious, but it is a little bit easier than, some of those other methods. This is what the seeds look like if it's really magnified under a microscope. They have this like very distinctive seed coat, and then they have a tail that often falls off when it's in the sand, but I think the tail might have something to do with how they like disperse by wind, even though they don't do a great job. Yeah, it's kind of like a little badminton birdie. So before I get into my statistics and numbers, does anyone want to take a wild guess at how much sand I processed for this study? Just throw out any number. I would love to hear it. Okay, that's definitely in the ballpark.
5 pounds.
Okay, getting warmer. 300.
300.
Oh, okay. The big reveal. 395. So it was quite a bit of heavy lifting for this project. And fun fact for any football fan, this is even more than William Refrigerator Bears. Iconic Bears defensive back in the 80s. So if anyone needs heavy lifting, you now know who to call. But let's get into the results now. So the first thing I looked at was comparing the mean seed abundance in the roadside and the rosemary balls. So this graph, it's just showing the mean seed abundance on the Y, and then I have my 2 locations on the X-axis. I did just a simple t-test for this one. And I did find significantly more seeds in the roadside. It's about like 3 in the road versus like 0.5 in the scrub on average, which really stood out to me because I would have thought that this disturbed area might have fewer seeds. So that was really interesting to see. Next, we're going into the rosemary balls. So the one on the left was looking at the effects of rosemary. So I looked at seed abundance for the far distance, so 2.5 meters away from rosemary, and then near distance, so 0.5 meters away from rosemary. I didn't find anything significant here. There's maybe a slight trend of finding more seeds farther from rosemary, but nothing super significant. But this graph on the right, so to analyze this, I did a generalized linear mix model where I accounted for presence of Eryngium as a predictor variable predicting seed abundance. And I did find a significant difference. So the presence of Eryngium does tend to predict where you will find seeds. However, as you can see, it's not like fail safe. I did find some seeds in plots where there wasn't any Eryngium, which also stuck out as an interesting result to me. This figure is more of a summary of all the different rosemary balls I visited. So I have seed abundance over on the y-axis and then the different rosemary balls on the x. So I want to put in the context of each of these rosemary balls before I talk about it. So the one on the far left is that extinct population that I went to see if it had any seeds. As you can see, I did not find any seeds in the soil there. But the most interesting thing about this graph to me was this second one right here, bald 54. So this is a really large population of Eryngium. If you've ever been to this area, it's just, there's probably more populations at this site than the others that I visited. But I didn't sample any seeds in that rosemary ball. And that really stuck out to me because it's like, there's a lot of Eryngium here. Why is there not seeds here? But at the other more medium-sized populations of Eryngium, I did find a lot of seeds. So now moving into probably my favorite figure. So this is my roadside analysis. So we're up at bulb 1, and along the x-axis, I have the distance going down the road. And I want to talk about the context of this bulb again. So this is a population that is somewhat newer. We think it was like started by scientists tracking seeds into this area. And the seeds started at 0, or the plants started more towards 0 and sort of like spread, invaded down this roadside. So I was very curious to see if that would, show a trend in what the seed bank is doing. And I made another generalized linear mixed model for this one. It was a negative binomial distribution because I had count data, but a lot of variance. So that model fit the best. And so we used distance along the road in this direction, the x direction, as well as this distance in the y direction as predictors for seed abundance. And it did, you can see it visually, but it did show a significant trend going down the road. So the seed banks are definitely becoming less abundant the further you move down the road, kind of following the invasion horizon, if you will, of that plant. It did also show a trend in this direction. It's a little harder to see from this figure, so I'm going to move to this figure. And just for context, since we've shifted our perspective, this is now the scrub line. So on the x-axis, I have that scrub to road distance. So all the places I took my soil cores, and then on the y, we have mean seed abundance. And this was found to be, the scrub to road distance was found to significantly impact seed abundance. We found this sort of sloped trend where at these intermediate distances of 2 and 3, there was a lot of seeds, but then as you get really close to the road, the seed abundance kind of drops off. So that could have something to do with like, maybe there's a certain level of disturbance this plant or like the seed banks can tolerate, and then it just becomes too much. And let me summarize all the conclusions here. So in terms of the natural populations versus the roadside populations, we found natural populations have a lower seed abundance than the roads. For vegetation, rosemary didn't really impact the seed bank. However, the presence of Eryngium did have a significant effect on the seed bank. And lastly, for the roadside, there were trends in both that X direction along the road as well as in the scrub to road direction. So for future directions, the first one is probably going to be the most important aspect of my project. So as I've been talking about plant lab demography a lot, but I think that having all of my like very detailed analysis of that roadside and then mapping on our demography data. So where are the plants, where are the new plants, and especially like where are the seedlings? So looking at, do those trends that we saw, do they match where the vegetation is? Do they match where the seedlings are? That's going to get at a lot of really interesting questions for this study. My second future direction. So this picture was taken when me and Andy were out collecting seeds for our plant lab seed collection work. And it was a very windy day. George was there too, actually. But we noticed that seeds were blowing all around the ground and they were sort of collecting where the sand had dips. And it made me start thinking about how these seeds are being dispersed. Like if they could have a very patchy sort of pattern as opposed to maybe a more even sort of pattern, which could also explain why like I might not have found seeds in samples I was expecting to. So I think there's a lot of interesting work to be done with dispersal. And this last picture is one of my favorite pictures. So George was kind enough to take me, Christine, and Shahram out to Hendry Ranch, where we found like the best population of Eryngium I've ever seen. It was like incredibly tall, tons of reproductive structures. Shahram is like, I can't believe it. And I'm like digging in the sand, like, are there seeds in here? Because what they do over there is they disc the soil pretty frequently. Don't know exactly the interval, but it got me to thinking like, what would the effect of disking be on the seed banks, and what would the effect of like different frequencies of disking be? There's definitely a lot of work to be done there. But two main takeaways for today's talk. The first one, I guess, is a little bit of a downer, but I feel like it's worth examining or just thinking about why these natural populations seem to have lower abundance, because it could have some implications for the populations of this plant. And this is a very endangered plant. So I think it's worth just noting as we continue to do demography on this species. And the second one is much more exciting, I think. So finding seed banks in the road is a really exciting thing, because it means that, you know, these plants couldn't just be like wiped out, like they are starting to establish themselves there. It's almost like Eryngium is discovering this new niche that it can use. And in terms of conservation, we could maybe use this to protect more roadsides or even start new populations in roadside. It's an interesting like case of coexistence, I guess, between an endangered plant. Yes, Jeffrey?
So build more roads?
No. Not quite. But yeah, it is a very interesting case that I think is pretty hopeful for this species. And of course, there's probably even more names that I need to include on this slide, but I have so many people to thank. Of course, Aaron, for being an amazing mentor. This is all super new to me. I've never done like field research before, and like having him help me from experimental design to stats analysis was amazing. Our RAs in the plant lab make it such a safe and welcoming space, and our interns do that as well. I've made such great friends here. I do want to shout out Ella for like talking with me as I began my project. She did sort of a past seed bank project, and we had to talk a lot about methods and things. Anyone who's done Eryngium demography ever, thank you, because I'm going to use your data. Julie for being like the best person ever, the smartest person ever. Kevin, thank you for pulling me and VT14 out of the mud one time. And yeah, I just want to thank, I do want to shout out the DEI working group. If anyone has questions on that, please reach out to me or Dustin or Katie, and everyone who just makes Archbold run, because this is a really unique place and a valuable program. So thank you so much, everyone, and thank you for listening. I want to open it up for questions. Yeah, Saha.
That was a fantastic talk.
Thank you.
The study was very well done. So do you think there are fewer seed predators along the roadsides than there are in the roadway points?
That's a really interesting hypothesis. I think there could be like, maybe because I don't know that much about animals, I gotta be honest, but I think like, I know a lot of animals do tend to forage more in those areas. So I think that's would be an interesting thing to investigate for sure.
I mean, I don't even know what.
I'm not too sure either, to be honest. We do find them herbivorized sometimes, but do you guys know what's usually chomping on them? I know they do experience herbivory from something. So that's a good hypothesis. Yes, Julie?
How did you move your almost 400 pounds stand back to the plant lab?
Well, okay, I didn't move it all at one time, but it sort of was an evolving process where first I tried to carry it in buckets, and that ended up in someone needing to come rescue me because I got so like heat exhausted. It was not good. But then Erin brought in this trusty little not little, but like a field like backpacking backpack, and I would pile all my sandbags in there and just like trudge back to my truck, because wet season also made this really difficult because I would often have to park my truck somewhere and then walk a good ways to do my sampling. So that was a challenge for sure. Yes.
Do you have any theories as to why you saw a low seed abundance and the site with the largest population?
That's honestly the thing that stumped me the most, because it did have a lot of very healthy plants. What I plan to do is map out, I didn't show it in this presentation because I'm still kind of working on it, but mapping out like the vegetation cover more in detail and like correlating that with the seed abundance. Like it could be that there was lower vegetation in those plots, but I don't have a guess beyond that as of now.
You're talking about like the seeds moving around or like
Yes.
sliding down the wall.
Right, it kind of just comes back to dispersal. Like there's so many ways they could be moving. Yeah, Hannah.
As far as the allelopathy with rosemary goes, do you know which plants do have a response to that?
I know Eryngium does have a response. Eric did a paper on that. Shown to have a pretty strong response actually on Eryngium. I think it might be mostly the herbaceous plants, but I'm not super sure beyond Eryngium. Yeah, Hillary? Oh, sorry.
I'm just curious about seed production
Mhm.
and to what extent, I don't know how many seeds you've counted on the heads of the Eryngium. It sounds like a terrible job. I suspect somebody at Oxford has done it. I was wondering if there was any spatial heterogeneity in seed production for those populations.
Yeah, I think that could come back to the demography data we have. We do count like flowering heads every time we do demography. I'm not sure how many seeds per flowering head we have, but I don't know, maybe like 10 per head. It varies a lot, but we definitely have the data on the number of flowering heads and like where the plant is. So I think we could definitely answer that question. It would definitely have an impact for sure.
A related question from Abe,
Oh, hi,
it
Ian.
was whether it was higher on the roadsides or the interior. I don't know. We definitely have that one in the data.
Yeah, it was definitely higher. At least the mean abundance was higher if I go.
On the individual plants.
Oh, on the individual plants. Oh, like the number of
There were
flowers
seed
higher?
production. Yeah. the seed production.
Oh, I think that's another question we could probably answer with our demography data. I don't know off the top of my head. To be honest, like just observationally, it might be higher on the road, but
Some of those plants are doing pretty well on the roads.
Yeah,
I wouldn't be
definitely.
surprised.
yeah. Yeah, Finny.
Was there any slope on the road that you noticed, or was it relatively flat?
I think it was a little bit sloped. I didn't measure it out, but it definitely like the road is like the low point and it goes up a little bit towards the shrub line. And we were thinking that that could very much affect like where the seeds are dispersing because they could be like rolling down the hill and sort of pooling at a certain location.
Yeah. Well, I think you answered this already, but someone named Dave Kennedy asked
Oh.
if any creatures are known to eat Eryngium seeds. I think
Yeah.
that was-
Yeah, we definitely know something eats them. I've never seen it myself, but we count herbivory for sure.
And Sharam asked, how does the spatial arrangement of Eryngium differ between roadsides and balls? Because of lack of cover or competition, or roadside populations denser?
Yes. Hi, Sharon. Good to see you. And absolutely, we definitely see that. They're just very dense on the road and definitely more spread out. I think my average like vegetation cover for in the balls was like maybe 4 or 5 plants per plot, and on the road it was a lot higher. So that's going to be a really important factor and predictor, I think. Yeah, let's see.
Did you identify all the different seeds you found or just Eryngium? And I wondered if there could be like more weedy species seeds in the road versus natural.
Yeah, that would be really interesting. I definitely noticed a lot of seeds as I was processing my samples, and there's like some very distinct ones. Like I'm not sure if I correctly identify this, but a lot of rosemary seeds, like the small spherical ones. That might not be rosemary seeds, but I definitely noticed other seed species there, and it would be very interesting to look at like if you could like sort of track all the different species you find there. I just looked at Eryngium because it was so easy to spot mainly. Yeah, Carrie?
How deep on average would you say your Eryngium seeds were inside of your sample?
So I only tested this in one rosemary vault. I threw some extra like not so great graphs at the end. But the one on the left was my depth analysis, and all of the seeds I actually found in that upper region. So like the depth of 0 to 4, I didn't find any in 4 to 8, but this is just one site. So I can't necessarily like extrapolate it to the rest of my data, but good question. Julie and then Finny.
Do you have favorite scrub plant? If so, why?
I'm so glad you asked that question. I've been thinking about this a lot. So Eryngium, obviously, but also I think I have to say Cladonia avancii. It's one of the ground lichens and it's very puffy. And if you've ever been out early in the morning and you've come across it, you can just squish it. And it's really great. And that's all I have.
We'll do one more question. This is from Dustin. How do you think researchers accidentally move the seeds to the roadside? Are these wind pollinated?
Yes, they definitely do disperse by wind. I don't know if people were just joking about this, but I think because this population is kind of further from the population, like other populations, people, at least like, I think Pedro always says like, oh, we track them in on our boots, something like that, because they are a little far from other Eryngium populations to have like wind dispersed all the way there, even though we can't necessarily rule that out.
Right. Well, I think we can let her off the hook from there. Let's give her a round of applause.